Welcome back friends, welcome back in another video tutorial from microbiology with Shatmik. In this video tutorial we will going to talk about the most widely accepted model to study the structure of cell membrane, that is fluid mosaic model of Singer and Nicholson. So let's get started with the video. So in the previous video we have discussed about the function of cell membrane, what are the utility of cell membrane in mostly prokaryotic cell, right? So today we will going to talk about the structure of cell membrane, and the fluid mosaic model, which was discovered by Singer and Nicholson in the year 1970s, was the most widely accepted model to study the structure of cell membrane. So let's see what points are included in that Singer and Nicholson model. So, first we have to know why the name fluid mosaic. So, the first term fluid is because the lateral movement of lipids and proteins. Okay, because you know what, the cell membrane, according to the Singer and Nicholson model, is made up of phospholipid bilayer, right? And in between them, proteins are present, right? So, fluid because Singer and Nicholson have observed the lateral movement of lipids and proteins. Then the term comes, that is mosaic. Mosaic because the mixture of lipid, proteins and carbohydrate. Because remember, cell membrane according to the Singer and Nicholson model is made up of three things: lipid, proteins and carbohydrate. Okay, so lateral movement meaning that if we consider these things are the lipid bilayers along with proteins, so they will move in this direction unless this direction. Okay, so that is the idea of lateral movement. So fluid because the lateral movement of lipid and protein, and mosaic because the mixture of lipid, proteins and carbohydrate. Okay, so that is the idea. So now let's talk about this model in a little bit more details. So first of all, the lipids which are present in the cell membrane according to Singer and Nicholson model, it is phospholipid. Okay, we have already done a lecture on phospholipid. You can watch this to understand the detailed structure of phospholipid. You may get this in the lipid playlist. Now, the proteins are of two types. One type of proteins are present in the periphery. They are known as peripheral proteins, and second one are integral. They are also known as transmembrane protein. We will going to talk about both peripheral and integral transmembrane protein in the next part of our video, where we will going to talk about the membrane proteins. It also contains carbohydrates, although the carbohydrates are in the form of glycolipids or glycoprotein. That means this kind of carbohydrate are attached with either lipids or proteins. Okay, so now there are three methods that help us to construct the model of cell membrane. So these three methods are transmission electron microscopy, fridge etching technique, and atomic force microscopy. So fridge etching, if you remember, it is nothing but the staining preparation of transmission electron microscopy. We have already talked about this in our microscopy lecture series. You can also watch this to understand, because here we will not going to talk about transmission electron microscopy or fridge etching or atomic force microscopy in details. We will going to just understand what are the outcome that help us to construct or confirm the Singer-Nicholson model of cell membrane, right? So first, transmission electron microscopy. So with the help of transmission electron microscopy, it was observed that after staining the cell, we are getting two darkly stained region, and in between them, or on either side of the interior, there are unstained regions. Okay, so that is the thing. These two things are darkly stained region, and in between them there is unstained region. Okay, that confirms that maybe the cell membrane is a bilayer. Okay, so it is near about 5 to 10 nanometer thin membrane, which was also observed with the help of transmission electron microscopy. Now, fridge etching. Fridge etching is the preparation technique for transmission electron microscopy. We have already discussed about this. So, fridge etching confirms that lipids are present as a bilayer, and in between them proteins are floating. And the last one, that is atomic force microscopy, confirms the previous two outcomes. That is how these three things, transmission electron microscopy, fridge etching and atomic force microscopy, help us to confirm that Singer-Nicholson model of cell membrane are mostly accepted model. Okay, so that is the thing. So this is how the cell membrane look like according to Singer and Nicholson model. There are phospholipid bilayers and in between them proteins are floating. Okay, so we will continue our discussion on Singer Nicholson model in the next part of our video, where we will going to talk about the proteins present in the Singer Nicholson. According to the Singer Nicholson model present in the cell membrane, they are peripheral and integral protein. Then in the next video we will also going to talk about the type of the lipids that is present in the cell membrane according to Singer-Nicholson model. That's how we will going to discuss about this Singer-Nicholson model. Okay, so that is it for today's video. I hope this video will be helpful for you. Thank you.